Today I'm going to talk about physical methods of sterilization. Sterilization is the process of destroying or eliminating all forms of microbial life, including bacteria, viruses, fungi, and spores. From a surface, object, or medium, it is widely used in microbiology, medicine, pharmaceuticals, and food industries to ensure materials are free from contamination. The sterilization can be achieved by four different types. They are physical agents, chemical agents, mechanical agents, and biological agents. And we're going to talk about physical agents today. Physical agents are of four types: dry heat, moist heat, filtration, and radiation. Mechanism of dry heat sterilization is different when it is compared to other types of physical agents, and dry heat works more slowly and requires high temperatures. So the mechanism includes protein denaturation, oxidative damage, and toxic effects of elevated levels of electrolytes. So here the protein denaturation. The protein denaturation, which causes denaturation and coagulation of proteins when the cell is exposed in a high temperature for a prolonged period of time. And the oxidative damage, which is caused to the essential molecules of like proteins, nucleic acids, lipids, and enzymes, and it disrupts the metabolism and structural integrity, leading to the cell death. And toxic levels elevated, which leads to the cell death. The imbalance of this concentration of electrolytes inside the cell leads to the cell death. The methods of dry heat includes direct flaming, incineration, and a hot air oven method. So the flame sterilization. In this, the objects are directly passed through a flame, usually a Bunsen burner, which burns and oxidizes microorganisms completely. So the instruments used here are inoculating loops or wires, points of forceps, scalpels, needles, or mouth of culture tubes, cover slips, glass slides, etc. are used here to sterilization. So the next dry heat method is incineration. In this, the complete burning of contaminated material are taken out at very high temperature, and the organism are oxidized and the material was destroyed to ash. The things that are used here are soiled dressings or animal carcasses or biomedical waste, which are turned to ash by incinerating. The most widely used dry heat method is hot air oven method. Here, the materials like glassware, forceps, scissors, syringes, swabs, liquid paraffins, or greases, fats are sterilized for 1 hour at a temperature of 160 degrees Celsius usually. Or the other temperatures used are 170 degree for 18 minutes, 180 for 7.5 minutes, 190 degree for 1.5 minutes are used. The next physical agent is moist heat. So the moist heat is more effective than dry heat because the water conducts heat better, and which allows fast penetration of heat into the microbial cells. So the mechanism here are denaturation and coagulation of proteins, like which results in the irreversible damage by the structural and functional loss, and it results in the cell death. And the second mechanism is the breakdown of nucleic acids. The heat and water damages the DNA and RNA, which prevents replication and transcription. So there will be a complete destruction of microorganism. And the next mechanism is the leakage of cell contents, which is caused by the moist heat. It alters the membrane permeability, and the leakage of cell contents results in the lysis of cell. So these are the mechanism which is performed by the moist heat. Methods of moist heat includes below 100 degrees Celsius, which is a pasteurization method, and at 100 degrees Celsius is boiling, and above 100 degrees Celsius, which is the steam under pressure. So below 100 degrees Celsius method, which is a pasteurization method in which there are two different types. The Holder method and the another one is the flash process. Here in the Holder method, the material is holded for 30 minutes for 63 degrees Celsius, and in the flash process, it is holded for 15 to 20 seconds at 70 degrees Celsius. So here there's a tabulation which gives the temperature and holding time for different microorganisms that are present in the different materials like milk or other wine or beer sources, which are used for the pasteurization method for the sterilization. So the next method is at 100 degrees Celsius. There are two different types. The one is boiling, the other one is steam at atmospheric pressure. First, we're going to talk about boiling. The next method is boiling method. Here in the boiling method, the vegetative bacteria are killed at 90 to 100 degrees Celsius. The spores are not been killed. For the effective boiling method, 2% of sodium bicarbonate is added. The hard water is not used for the boiling. So this is the boiling method which is not used for the destruction of spores. It is only used for the destruction of vegetative bacteria at 100 degrees Celsius. So the next process is the steam at atmospheric pressure method. It uses a Koch and Arnold steamer, which is an inexpensive method. This uses 100 degrees Celsius for 20 minutes holding time and temperature for 3 successive days, which is known as sterilization. This intermittent sterilization kills the vegetative bacteria on first exposure, and the spores present in the medium being in a favorable condition and will germinate and be killed in other consecutive exposures. All the spores cannot be killed like anaerobes or thermophils, but most of the spores can be killed. So the next method is above 100 degrees Celsius, which is a steam under pressure. It is an autoclave method in which 121 degrees Celsius at 15 psi for 15 to 20 minutes the surface of the media is sterilized. The steam penetrates into the material and it causes rapid protein coagulation and spore destruction. So in short, like moist heat kills by denaturation, coagulation, and hydrolysis of proteins and nucleic acids, along with disruption of membranes by above 100 degrees Celsius, at 100 degrees Celsius, and below 100 degrees Celsius. The next physical agent is filtration, which is a mechanical method of sterilization where microorganisms are physically removed from liquids or air by passing them through a filter with pores. So the mechanism here is size exclusion and sometimes maybe adsorption. Here the microorganisms larger than the filter pores are trapped on or within the filter. There will be a different pore sizes, for example 0.45 micrometer which removes bacteria, or the 0.22 micrometer which removes even smaller bacteria and spores. So the ultrafiltration, which is 0.01 micrometer or less, can remove viruses. So the types of filters are candle filters, asbestos filters, sintered glass filters, and membrane filters. So the first type is candle filters. Candle filters are used for large volumes, but it may allow viruses to pass. And then the best candle filter is the Chamberlain and Backfeld filters. So there are two types of filters: unglazed ceramic filters and diatomaceous earth filters, which is made from porcelain or diatomaceous earth material. Is asbestos filters, which is a single-use and disposable filters, which is not a common filter because it was a carcinogenic material. It may be hazardous to the medium. So the next type of filter is sintered glass filter, which is made of finely ground glass particles fused together and is less common due to the fragility. The next type of filter is membrane filter, which is made of cellulose esters, nylon, or polyesters, polybicarbonates. The pore size of the membrane filter is 0.22 and to 0.45 micrometer, which is a commonly used filter in microbiological laboratories for sterilizing antibiotics, serum, vaccines, enzymes, or urea broth, etc. Physical method is the radiation, which uses energy waves like electromagnetic waves or particle radiation to kill or to inactivate microorganisms by damaging their DNA, RNA, and essential biomolecules, their metabolism. And there are two types of radiation. They are non-ionizing radiation and ionizing radiation. First is non-ionizing radiation, which is an ultraviolet radiation between the wavelength of 220 to 300 nanometer. The mechanism of this UV radiation is that it causes thymine dimers in DNA, which prevents replication and transcription and leads to the mutation and death of a microorganism. So the next is ionizing radiation, which is gamma rays from cobalt 60 or cesium 137 also, and X-rays and electron beams, which ionizes water and other molecules and produces free radicals. So these free radicals causes DNA breaks, protein damage, and membrane disruption for microorganism and leads to the death of a microorganism. So these are the physical agents which are used for sterilizing medium or the instruments used for the experiments. So thank you.